Hello, my name is Brian Duff. I am Technical Lead for the PSC portfolio. Welcome to PSC Foundations Module 9, Characterization and Cell Quality of hPSCs. After this module, you should be able to outline the characteristics of hPSCs, understand the importance of assessing hPSC cell quality, discuss assessment of hPSC quality with respect to proliferation, morphology, pluripotency, and genetic stability. This module contains 4 sections. Section 1, Characteristics of hPSCs. Section 2, The importance of cell quality in hPSC culture. Section 3, How to assess cell quality looking at different characteristics of hPSCs. And Section 4, the summary. Sidebar sections will highlight applications and other useful information related to the curriculum. Section 1, Characteristics of hPSCs. hPSCs possess the ability to self-renew and proliferate. They exhibit normal cell and colony morphology. They have high expression levels of markers of undifferentiation. They are capable of differentiating into cells of the 3 different germ layers, the ectoderm, the mesoderm, and the endoderm. They have normal human karyotype. And now we will go through each of these characteristics one by one. So let's look at their ability to self-renew and proliferate. With in vitro culture, most primary cells will only divide for a finite number of cell divisions before they begin to senesce or stop dividing. They have a finite number of cell divisions. In order to keep these primary cultures growing indefinitely, they usually have to be transformed into an immortalized cell line with a virus or an oncogene. However, hPSCs have the ability to self-renew and grow indefinitely without the need to be transformed. In the right culture conditions, hPSCs can be expanded while maintaining undifferentiated features. In vitro, the environment must be conditioned in order to mimic the in vivo environment. Therefore, a substrate to attach to and nutrients, as well as the homeostasis of the in vivo environment, are mimicked in vitro with a matrix, specialized media, and the incubator, respectively. Being pluripotent, hPSCs can inherently differentiate. So upon cell division, the progeny may either be a copy of the parent hPSC or a differentiated cell. Therefore, in culture, even when the correct pluripotency factors are available in the media, it is normal to have a low percentage, anywhere from about less than 10 to 15% of spontaneously differentiated cells present. If the percentage increases far beyond this, the researcher should be concerned about the quality of the culture. Now let's look at what is considered normal cell and colony morphology. As in the top image, under high magnification, hPSCs have a high nucleus to cytoplasm ratio, seen here with the blue arrow, and prominent nucleoli, depicted with the orange arrow. The bottom image shows the appearance of normal morphology of hPSCs at a low magnification. The colonies are relatively round with defined borders. As the colonies grow, the centers become multi-layered, causing them to appear bright when observed under a phase contrast microscope. hPSCs express markers of undifferentiation at high levels. Markers of undifferentiation are expressed at high levels in healthy hPSC cultures compared to differentiated cultures. Common markers that researchers look at include Tra160, Tra181, OC3 and 4, SSEA3 and 4, SOX2, and NANOG. Please note, we call these markers of undifferentiation rather than pluripotency markers, as some of these markers are also expressed on other undifferentiated cells, such as cancer cells. This is why it is important to look not only at marker expression, but also other characteristics of hPSCs when assessing cell quality. Pluripotency is a functional description of the cells and can only be shown by differentiation. The graphs on the right compare relative marker expression of OCT4 and Tra160 across different ES and iPS cell lines when cultured in M-teaser1 and M-teaser plus. Marker expression is very comparable in both media. These analyses were carried out by flow cytometry, which is quantitative, but other methods such as Western blot, which is semi-quantitative, or immunocytochemistry, which is qualitative, are also commonly used to assess protein expression. Alternatively, RNA expression can be measured by RT-PCR, qRT-PCR, or Southern blotting. hPSCs are capable of differentiating into cells of the three different germ layers, the ectoderm, the mesoderm, and the endoderm. By definition, hPSCs are capable of differentiating into all three germ layers. This is what pluripotency means. This can occur spontaneously or through directed differentiation and can be done either in vivo or in vitro. The in vivo gold standard for assessing functional pluripotency of a cell population is the teratoma assay. Here, hPSCs are injected into immunodeficient mice, and the teratomas that form are removed and analyzed for the presence of cells or tissue from all three germ layers. However, there are many drawbacks. Depending on the paper cited, there is inconsistency with respect to the injection site, the number of cells implanted, the vehicle for injection, the age and strain of the mouse, when to harvest the cells, etc. As well as this, many teratomas can become cystic and difficult to analyze, and there's always a requirement for an experienced pathologist who can identify all different possible cell types, making this a very costly experiment. The image on the right, taken from Damianov and Andrews in 2016, illustrates a teratoma slide exhibiting muscle, cartilage, neural tissue, and endodermal tube structures, respectively. Because of the drawbacks associated with the teratoma assay, researchers are moving towards in vitro alternatives. One method to induce spontaneous differentiation in vitro is to aggregate the hPSCs into 3D non-adherent structures called embryoid bodies, or EBs. This structure is thought to mimic the 3D spatial arrangement of the developing embryo. When supplemented with non-specific differentiation conditions, as in with the use of serum-containing medium, the cells within the EB will spontaneously differentiate. The EBs are then plated down, and the outgrowths are stained for the expression of lineage-specific markers to detect all three germ layers. Alternatively, the differentiated cells are analyzed by qPCR for lineage-specific markers. It is common for this type of differentiation to result in higher amounts of neuroectoderm cells, as these types of cells are the easiest cells to differentiate too. But hPSCs are functionally pluripotent. Cells of all three germ layers should be present. The EB differentiation assay can be quite variable due to many factors, such as the size of the EB generated, the lot-to-lot variability of serum, and the duration of the assay. Due to the variability with EB-based spontaneous differentiation protocols, in vitro directed differentiation protocols have been developed whereby specific cytokines and small molecules are added to media to direct the differentiation of hPSCs to specific cell types. Using different cytokines and small molecules, hPSCs can be separately induced to differentiate into neural progenitors, mesodermal progenitors, and definitive endoderm cells, thus proving the pluripotency of the original hPSC culture. The progenitors can be further differentiated and matured into terminally differentiated cell types if required. The graph on the right shows relative expression of ectodermal, mesodermal, and endodermal markers expressed in cells which were directed to each respective lineage separately following maintenance in either M-teaser1 or M-teaser plus. hPSCs have a normal human karyotype. Normally, human cells have 23 pairs of chromosomes with well defined arm lengths and banding patterns, as visualized by karyotyping, also known as G banding. A karyotype is simply a picture of a person's chromosomes. To obtain this picture, the chromosomes are isolated, stained with Giemsa stain, hence the name G banding, and examined under the microscope by trained cytogeneticists. If the karyotype becomes abnormal over time, for example, an extra chromosome present in trisomy 12, then the quality of the cells has been compromised. Of course, this is for normal cell lines. There are lines that have abnormal karyotypes and are used for disease modeling. For example, cell lines with the Down syndrome karyotype. Although karyotyping can detect gross genetic abnormalities, detection of smaller aberrations may require more sensitive methods of detection. For example, fluorescence in situ hybridization, or FISH, whole genome sequencing, or qPCR. The image on the right shows a normal human karyogram for the iPSC line WLS1C. Section 2, the importance of cell quality in hPSC culture. So why do we care about hPSC quality? Well, biomedical research is in a reproducibility crisis. The image on the right, taken from Friedman et al. In 2015, shows that 50% of the US annual preclinical research funding is spent on research that is not reproducible. This is particularly poignant for the hPSC field, where there is still no consensus on what quality control checks are required in order to publish. Additionally, as more clinical trials containing hPSC derived cell therapies are entering the clinic, there needs to be a consensus for characterization and cell quality attributes of hPSCs and their cell therapy products. For example, in 2015, Illich and his colleagues reported on a Japanese clinical trial for iPSC derived retinal pigment epithelial cells, and this trial being suspended when autologous iPSCs from a patient did not pass a genomic validation step due to the presence of an oncogene. We need to be sure that the cell product we are delivering to a patient does not contain oncogenic material. Numerous consortia are working hard to define the role of these guidelines, and STEMCELL is working closely with them on this. In 2016, Susan Solomon and her colleagues at the New York Stem Cell Foundation raised this issue with the paper entitled Raising the Standards of Stem Cell Line Quality. This publication drew significant attention to the issue and raised some very important points as seen below. Section 3, Assessment of hPSC Quality. STEMCELL is invested in the idea to standardize and promote the ideals of hPSC quality. When we talked to our customers, it was difficult to find a consensus on what exact attributes of hPSCs defined quality, even within the same lab. However, some recurrent terms kept coming up again and again in these customers' discussions. We have adopted these terms as the pillars of hPSC quality. They are pluripotency, genomic instability, morphology, epigenetic landscape, and gene and marker expression. Although the field is aware that the epigenetic landscape of the cells must play a role in their overall quality, it is still unclear how this is the case. At this time, we will discuss the other 4 pillars and how they should be assessed to give an overall picture of hPSC quality. In order to assess hPSC quality, we need to assess proliferation, cell and colony morphology, pluripotency, and genomic stability. Let's begin by how to assess proliferation. It is now known that some genetic aberrations that occur in hPSCs confer a growth advantage to the cell. Therefore, if a cell line is passaged on a consistent basis, for instance every 4 to 7 days, and reseeded at a consistent split ratio or clump number, but a change in growth rate is observed, we recommend that the customer assess other cell quality attributes such as morphology, genetic stability, and pluripotency to further assess the quality of the cells. Clump passaging is recommended as it has been shown that hPSCs that are routinely passaged as single cells may more easily acquire certain mutations that confer a growth advantage. Assessing cell and colony morphology. As mentioned previously, healthy PSCs should display round, tightly packed cells with a high nucleus to cytoplasm ratio. They typically have smooth defined colony borders and are relatively circular, but this can depend on the matrix. There should be multilayering at the center of the colony at the time of passage, visualized as phase brightness on a phase contrast microscope. This is an easy assessment to do, it can be done daily, and the growth of the culture can be easily tracked. However, it is somewhat subjective. One tends to get better at determining the quality of the culture based on morphology with experience. The morphology can also differ depending on the culture conditions or cell state, i.e., feeder versus feeder free conditions or naive versus primed PSCs, for example. However, morphology is a sensitive indicator of cell quality. In this experiment, hPSCs were cultured in M-teaser1 and a suboptimal medium, which was M-teaser1 without some pluripotency factors, for 5 passages. Using an R&D based morphology scoring system, the quality of the cultures were examined at each passage. Surprisingly, it wasn't until passage 4 that a drop in the morphology score of cells in the suboptimal medium was noted. At passage 4, this medium exhibited spiky colony edges, loosely packed cells, and excessive multilayering. This low score would account for a fail in R&D testing. Surprisingly, at the same passage, some cells were examined for markers of undifferentiation. Expression levels of Tra160 and OCT4 were still high, indicating that changes in morphology are often seen ahead of changes in marker expression levels. Practically the entire well would need to appear morphologically differentiated before one would expect a significant drop in the well's marker expression. Assessing pluripotency. As mentioned earlier, although the teratoma assay has been known as the gold standard for assessing pluripotency, due to the drawbacks associated with it, researchers are moving towards the use of alternative in vitro methods. Some quantify expression levels of markers of undifferentiation. OCT4, NANOG, and SOX2 expression all correlate with the teratoma forming potential of hPSCs. It is important to remember that these are not markers of pluripotency and can be found on other undifferentiated cell types such as cancer. Although, as seen previously, changes in marker expression may be noted after more evident changes in morphology appear. Some researchers choose to use computer prediction models such as Pluritest or Thermo Scorecard. Pluritest is a bioinformatics assay in which the transcriptome of a test cell line is compared to the transcriptome of a large number of cell lines known to be pluripotent. Thermo Scorecard uses a panel of 96 genes that identify the differentiation capacity of a given cell line more quantitatively than the teratoma assay. However, more researchers are carrying out both spontaneous and directed differentiation protocols in vitro to determine pluripotency potential. Arrays for genes of undifferentiated versus differentiated cell types are also becoming popular in assessing pluripotency. In the heat map on the right, you can see that undifferentiated H9 ES cells expressed high levels of hPSC associated markers. EBs generated using a 10-day protocol in serum containing medium did not show significant upregulation of most mesoderm and endoderm markers. As mentioned earlier, this is not uncommon in spontaneous differentiation protocols, as it is easier for many hPSCs to differentiate to ectodermal cells more so than mesoderm or endoderm lineage. However, following directed differentiation using our STEMDiff trilineage differentiation kit, there was clear upregulation of lineage-specific markers in samples directed to those lineages respectively. Assessing genomic stability. In culture, hPSCs can acquire recurrent karyotypic abnormalities. This is due to the artificial environment of the culture dish, which subconsciously biases towards a specific phenotype. Following prolonged culture in vitro, we unwittingly select for the cells that have a decreased population doubling time, i.e., the ones that grow faster, and the ones that attach easier and have increased plating efficiency. These phenotypic aspects can be linked to karyotypic changes observed by G banding. Also, some karyotypically abnormal cells display a growth advantage. The danger is that a number of these recurrent abnormalities are also observed in a number of human cancers. Therefore, in the realm of cell quality, we need to be sure that we are culturing hPSCs and not cancer cells. It is really important to routinely assess the genomic stability of hPSCs in culture. However, complete genome analysis can't be achieved using just one method. Also, every genomic analysis method has pros and cons. The turnaround time can be quite different also. Right now, each lab or institute determines which analyses should be employed and how often. This table outlines some of the most common techniques, their sensitivities and detection limits, and the typical turnaround times. G-banded karyotyping will detect most inversions, duplications, deletions, translocations, and aneuploidies, i.e., growth abnormalities. However, it will not pick up submicroscopic abnormalities, i.e., those that are less than 5 megabases in size. The detection limit in the sample population needs to be greater than 15%. In other words, the abnormality needs to be present in greater than 15% of the cells in the sample, and the turnaround time is about 1 to 2 weeks. Microarrays such as comparative genomic hybridization or single nucleotide polymorphism arrays will detect copy number variations, duplications, deletions, unbalanced translocations, and aneuploidies. They can't detect balanced translocations and inversions or the chromosomal position of genomic gains. The detection limit in the sample populations here needs to be greater than 10% to be picked up, and the turnaround time is about 2 to 3 weeks. Fluorescence in situ hybridizations, or FISH, can detect a genomic sequence of interest, but it won't pick out changes in regions other than the sequence of interest. It is quite sensitive. The detection limit in the sample is as low as 5%, and the turnaround time is similar to that of karyotyping, so about 1 to 2 weeks. And whole genome sequencing can detect duplications, deletions, aneuploidy, balanced translocations, inversions, and single nucleotide variations. Because you're looking at the whole genome, the analysis is very complex and probably prohibitively expensive to carry out routinely. The turnaround time is about 3 to 4 weeks. So, with all of the different methods for assessing genomic stability, once the lab has decided on which methods to use, how often should this be done? Customers ask this question all the time. You analyze the stability and ensure you have a normal karyotype, then you culture your cells for X number of passages, and all of a sudden you have an abnormality such as trisomy 12. So, should you do this once every 10 passages, once a month? Really, it's based on the feasibility in the lab, but once every 10 passages is a pretty good benchmark. It's also important to keep high quality, genomically stable cell banks so that if or when an abnormality happens in culture, you know what passage to revert to that is known to be genomically stable. One tool which can be used and will be discussed in more detail in the PSE product modules is the hPSC Genetic Analysis Kit. This kit is a qPCR assay with probes targeted towards the minimal critical regions on 8 recurrent abnormalities known to occur in hPSCs. The kit was designed based on work carried out by Peter Andrews and Ivana Barbarich's team in Sheffield, of which Adam Hurst is an author. A region on chromosome 4 is used as a negative control region as it is highly conserved. The kit will probe approximately 75% of reported abnormalities in hPSCs. One of the genetic assays probes for trisomy 12. The result of the genetic analysis kit can be seen on the left, where an additional copy of the chromosome is present. The confirmation of the result was carried out by karyotyping. The kit also detects submicroscopic recurrent abnormalities such as 20q1121 duplication. This aberration can sometimes be detected by G banding depending on the size of the amplicon. However, it is often missed. Therefore, confirmation of the increase in copy number on the left was done using FISH. The image on the right clearly shows a cell containing 3 copies of 20q1121. So, in summary, characteristics of hPSCs include the ability to self-renew and proliferate indefinitely under appropriate conditions, the exhibition of normal cell and colony morphology, high expression of markers of undifferentiation such as TRA160, OCT3 and 4, SSEA3 and 4, and NANOG within a culture, the ability to differentiate into cells of the ectoderm, mesoderm, and endoderm, and possession of a normal human karyotype. The importance of hPSC quality is paramount given the advent of hPSC-derived cells in cell therapy. This requires a consensus for characterization and cell quality attributes of hPSCs and their subsequent cell products. At STEMCELL, we recognize 5 pillars of cell quality: morphology, pluripotency, gene and marker expression, genomic integrity, and epigenetic landscape. Many of these attributes should be routinely assessed to determine the overall quality of the hPSCs by all researchers working in the field. Thank you for your attention.